I'd like to thank the organizers for inviting me to this wonderful workshop. So many wonderful talks and stimulating conversations, and still more to come later this week. All right, today I will present some of our recent work on actually network data integration or network data fusion. So this is just an overview of my talk. I will just briefly introduce the problems. I mean, they were introduced in many talks, so I'm not going to focus on that. And then I will present one methodology, basically, that we've used over the past 3 years or so to fuse various types of biological data, network and other types of data, and to do various tasks, for example, to reconstruct Go, to find patient-specific things such as stratification, drug repurposing, and biomarker discovery, to reclassify diseases based on the evidence from multiple omics-type molecular and other data, and in the end, to do multiple network alignment. And in the past, we focused mostly on single network analysis, and I'll just briefly basically say how that contributes to these tasks. All right, so let's start with a brief introduction. And this doesn't work. Why doesn't this work? Okay. So we all know one of the most complex systems that we study is the cell, and in the cell there are many different types of networks. For example, there are many types of networks between genes, whether it's co-expression or transcriptional regulation and so forth. Then genes make proteins, and proteins can physically bind to do particular tasks. So we have protein interaction networks. Then we also have various types of networks on diseases. For example, we have disease ontology, or we can link diseases if they are comorbid in particular patients and so forth. Also on drugs, we can have various networks, whether it's drug chemical similarity or whether drugs are given to the same patient group and so forth. So basically, we have all of these network data types, co-expression, protein interactions, various genetic interactions, metabolic networks. Then we have various ontologies such as disease ontology, gene ontology. Then we can have drug side effects. We have so much data, and that basically there are questions of how much information we can take out of each of these data sources individually, and then how we fuse them all together, basically to see what they collectively tell us. And why? This is because all of these data sets give us different views of the same system of the living cell that complement each other. And the question is how to analyze them collectively, because only by collective analysis we can get a full insight. All right, but as we all know, it's not trivial to deal with these problems. Even just a simple thing such as comparing networks, it's already a hard computational problem, and we need to rely on heuristics. And basically throughout the talk, I'll point to basically NP-hard problems for which, of various types, for which we design heuristics to try to fuse these data to learn more, basically to mine information collectively from these data types. Okay, so now about the methodology. And first, basically, I will lead you through these 4 examples. These are our 4 recent papers, and just basically going from the simpler model towards a more complicated model and how we've done it. Okay, so let's first talk about our paper from ECCB from a couple of years ago, how to reconstruct Go from molecular network data. And Trey gave yesterday an overview of his work on this. So basically, we were inspired by his 2013 paper, basically that asked how much of can we reconstruct Go from the wiring patterns of various types of data. And unlike Trey's approach, we used the semi-supervised co-clustering machine learning method. And that method learns from the relation patterns both within the same type, and the types that we fused here are PPI network, genetic interaction network, co-expression network, and yeast net. So this is yeast. Okay, so one data types is basically genes or gene products, proteins, and various types of interactions on them. Another data type is Go terms. Okay, so we want to simultaneously basically co-cluster these 2 data types based based on the evidence from all these data. So our method learns from relation patterns both intra-type, within one data type, and across different types. So this is across these annotations, and predicts new gene annotations. So basically these links between Go terms and genes, but also it infers new Go term associations from co-clustering, by co-clustering, basically within the same framework. And just a result that I'll come to at the end, that we can actually recover 96% of Go annotations from fusing basically just these topologies of these data. All right, so how do we do that? So we want to simultaneously cluster Go terms and genes. And the motivation comes from non-negative matrix factorization, which factorizes a non-negative data matrix. Okay, and this is homogeneous data. So this is one data type. So it factorizes this non-negative data matrix into two matrix factors. Okay, and this is an alternative to principal component analysis and vector quantization. So the theoretical foundation for NMF for clustering homogeneous data is that it is equivalent to a relaxed form of k-means, where this factor contains the centroids, cluster centroids, and this factor contains cluster membership, and k is the number of clusters. Okay, and this is basically from this paper. That this is not our work. This is from 1999. Okay, but this is homogeneous data. This is the same data type, but we want to cluster Go terms and genes simultaneously. Okay, and this is why we have to use non-negative matrix 3 factorization, and it's penalized or graph regularized to include these penalty terms that come from the topology of these networks. And how do we do that? Okay, so this matrix R12, which is basically Go term annotations. This is relationships between genes and Go terms. This is R12. We approximate it by these three matrix factors. Okay, so it's penalized, and I'll show you in the next slide. Basically, it integrates topologies of molecular networks as constraints, so that's why it's semi-supervised. It's non-negative, so that we would have cluster interpretation of these estimated matrix factors. And it's 3 factorization because it simultaneously clusters columns and rows, genes and Go terms. And over here, K1 is the number of clusters here, and K2 is the number of clusters here in Go terms. Now, how do we compute this? So we are approximating, again, we are approximating this by these 3. How do we do that? So we do that by solving a continuous optimization problem, which minimizes this. So R12 minus this here. Okay, this is it. This is Frobenius norm. But under the guidance of pairwise constraints that come here. Okay. Now, in these constraints, so this is basically, these are the Laplacians, this L1 are Laplacians of these 4 matrices. And the idea here is that basically in these Laplacians you would have they will have a negative value if there is an edge, okay? And this will be a reward to your function. Basically this will, you want to minimize, so this will lead your function towards a local minimum, of course, because this is an NP-hard problem, so we can only approximately find these local minimum. Okay, so this is I1 to 4. These are basically the topologies of these 4 networks. Now, these 4 here are the topologies coming from graphlet similarities that Igor just mentioned. So basically, while this takes into account direct connectivity, this takes into account similarity in the wiring patterns of faraway nodes. And why is this? And I will come back to this slide. I'll just remind you that basically, as Igor said, as Igor said more than a decade ago, I introduced these graphlets. Kind of the idea was that these are Legos, kind of like building blocks of these networks, and we would figure out by the way that they are linked to each other via these basically symmetry groups in them, or automorphism orbits. So basically the way or these correlations by which they come together would tell us whether these large data, whether these Legos make up a pumpkin or an airplane or whatever that is. But I will not talk about graphlets in this talk. I'll just like to remind you that we have already shown a long time time ago that similarity in the wiring patterns far away nodes means statistically significant similarity in the biological function, membership protein complexes, subcellular localization, tissue co-expression, involvement in disease, and our predictions of new melanin members of melanin production pathways were phenotypically validated by siRNA screens at UC Irvine Dermatology Department while I was an assistant professor there. And more recently, we've come up with a machine learning method where we linked basically these orbit positions with different GO terms, and we have shown that certain functional groups tend to have preservation in the topology even for the faraway species. And of course, there is still more improvement on this that we haven't published yet. And the work on graphlets, we have done some work by which we can track the network dynamics, so basically how the structure of the network changes, and everything is recently generalized to directed network, and we just put this on archive a couple of weeks ago. But let's get back to this topic. This is why we have this term here coming from all of these data, and this is, so again, and now our matrix is graphlet degree vector similarity, and then this is just the same stuff there, but with basically to capture that. And also we add this term not to ignore, basically we want to, we don't want to ignore what we already know from Go. So we put that in, we can put that out, but yeah, in this paper we put that in. Okay, so now how do we minimize our objective function? We decided to follow multiplicative update rules obtained from KKT optimality conditions. There are many ways to do that, but this is just one way we did that. So how do we do that? We basically randomly initialize these matrices G1 and G2. Then we follow these update rules. So for every model, you see that there are different models. You have to derive these, because we are dealing with NP-hard continuous optimization problem, and the update rules are like this. So, you know, this is how you update S1-2, and this is how you update G1 and G2, and your stopping criteria, basically you stop when your functions, you know, when the difference is too small. And again, this is not ours, this is from the paper from 2008. Okay, right. So now we know how to approximate this with these three matrix factors. And then we are obtaining knowledge out of these factors. Because there were random initializations here, then we have to run it a few times and to derive a consensus matrix. So basically, for example, if we want to figure out which Go terms are together, we go along here, along this row, and then define the maximum one. We construct, this is in our ith run CI, and then in the end, we average over that to derive this consensus matrix, and then we have these clusters of Go terms. And R1-2 you basically reconstruct directly, so to recover existing and predict new gene annotations. Okay, so now some results. The beauty here is that you can remove any type of data and see how much degradation to your model you get. And we, in this particular case, we evaluate model by residual sum of squares and explain variance. And we see here that just as Trey mentioned yesterday, that if you remove genetic interactions, then it's the largest degradation. So genetic and PPI interactions are more relevant than co-expression data. We can argue why that is. And also we tested whether graphlets contribute. And yes, if you remove the graphlet part, also your model degrades. Now,
the whole data set.
Entire data set. You remove genetic interactions, all of them at the time, and you keep these. And you will see in later examples how basically, you know, by integrating more and more data, how your accuracy of the model improves. And that's why this is nice. New associations between Go terms. So basically we want to figure out how much of this holds in the light of the evidence from all data. And basically, we find that the overlap between the Go term classes that we obtained and the actual Go is 96%. And then we predicted Go term associations that are currently not present in Go. We predicted 132 of them that are statistically significant, and we validated 58 of those through literature curation. Now, in terms of new gene annotations.
Your model, you are using Go as one of the sources. So
I am using here Go as one of the sources. You can exclude that, yes. Yes. Yes.
excluding all Go.
The evaluation is done by excluding all Go. I don't remember. let me look it up. I'll get back to you.
Also, 96% doesn't tell us.
It doesn't. No. It just tells you exactly. Yeah. So it's an imperfection of this method for this particular application. Yeah. So in terms of new gene annotation, so basically now we are talking about this R12. We do a threefold cross-validation. We remove a third of the data, and this is the area under the ROC curve. This is 87%. We evaluated, so whether we do correct assignment of Go terms to particular genes, and we find out that yes, we do that, and graph degree vectors improve results, and that we outperform what Trey talked about yesterday, and we can do that for a cellular component, biological process, and molecular function, and the red one is when you include graphlets as well. And we actually do biological validations of these predictions by using Charlie Boone's new genetic interaction data that is available now online. And basically, we see, that the distribution of genetic profile correlations amongst new annotated gene pairs significantly differs from the distribution of the randomly sampled ones. And we use Kolmogorov-Smirnov test to do that. All right, now let's go to the title. So basically, this was the first example because it's the easiest to describe the methodology, because we co-clustered only genes and Go terms, two different types. Now we want to simultaneously cluster patients, genes, and drugs, and to do these particular tasks in precision medicine. And this is actually our recent paper that appeared in PSB this year. Okay. Right, so now it's patient-specific data fusion. We go to TCGA, and we got about 450 ovarian cancer patients. And as I said, we have three entities, three different data types: patients, genes, and drugs. We have the somatic mutations profile, so from each individual patient, we know which genes are mutated. Okay, on genes we go to BioGrid and KEGG. We have protein interactions, genetic interactions, metabolic interactions. Okay, so this is our R12 matrix. Then we go to drug bank, and we take the data about drugs and their targets. Okay, here. And then we have drug chemical similarity network here, and we used SMILES there. Okay, so this is where the data comes from in this particular example. Now, this R23 matrix is between these two data types. And again, we are doing the same thing. We are minimizing this objective function, where R12 we are approximating by these matrix factors, R23 by these ones, but notice that we share D2. And by sharing this matrix factor, this is how we ensure fusion of the data. And then this is, again, the same as before, penalties coming from the topologies. So this is how that works. Okay, basically for drug repurposing, we want to reconstruct some of the new of these R23. This comes directly from R23 here. Patient stratification comes from this factor. So this is clustering of patients based on evidence of all of these data. And driver gene prediction, this is the biomarker we wanted to figure out whether we can do something with it. So driver gene predictions comes with this from this G2, and this is those consensus matrices that I told you about because of this randomness in these experiments. Some results, overview of the results. I don't have much time, less than 5 minutes left. So in terms of patient stratification, this method gives us three groups of patients with statistically significantly different survival curves, Kaplan-Meier survival curves, and basically.
It's something that you pre-specify for them.
There is, so basically, when there is those K parameters, then you play with them. Okay, so right, and then it's not something... that's the one that gives the best clustering. Yeah, again, another think of the methodology. Yes. Okay. So that's in terms of patient stratification. In terms of predicting driver genes, basically it comes from here, and we see that the kind of the functions that are involved in cell proliferation, progression, anti-apoptosis, and so forth. Now we predicted 109 new driver genes. But 40% of them we validated in databases that we didn't use in this particular fusion experiment, right? So this is where the validations come from. And this is what I told you. This is how I will show you how basically your accuracy improves as you add more data. For drug repurposing, basically reconstruct this matrix. This is when you only take drug-target interactions to infer new drug-target interactions. And there is red is the area under the ROC curve, and this is area under precision recall curve. And basically, as you add more data, you see that basically with the addition of all data, it's the best performance of the model. Okay. And we predicted many new drug-target interactions, and then 37% of them we confirmed in basically the databases that we did not use here. Okay, this is a work in preparation. We work with Professor Stabbing's lab at Imperial. This is a small set of cancer patients, of 45 patients, and this is just an illustration how all cancers are different. So we have, this is hard to read, I'm sorry about that. So this is our 45 patients, and this is basically how many genes are mutated in them, and we see that most patients have very small number of genes mutated, like 5 to 10. This is genes. There is around 180 genes mutated in these 45 patients, and we see that a particular gene, these are particular genes, a particular gene would be mutated in a very small number of patients unless it's TP53, which is mutated in less than a half of patients. So very, very versatile disease. So we have these patients, we have drugs, we have genes. Again, you have different networks here. You have different relations between different data types, and you want to use them all to predict which drugs, already approved drugs, should be given to which particular patients based on all of this, based on their mutations and evidence of all data. And again, I would like to stress that here basically you're decomposing all of these, but you're sharing these matrix factors, and this is why this works. So this G1 over there, G3 over there, and G2 right there. Okay. And I don't have much time, but I just like to say that we did also disease reclassification. Basically, the disease ontology, the way doctors learn it now, is about 100 years old and based only on the symptoms and the most affected organs. And the question is how much of that classification still holds in the light of new molecular level data that did not exist 100 years ago. And we've all seen these kind of projection methods, and we know there are limitations, and we actually did, again, similar type of fusion. But again, I need to stress that each of these, you have to different multiplicative update rules, or you have to decide how to converge, and these are non-trivial problems, and they're compute intensive as well. So over here, the model is the most complicated in the sense that you now have 4 different types of objects. You have genes and different types of interactions on them. You have drugs and different types of drug interactions on them. You have gene ontology and disease ontology, and you are fusing it all as before. You get all of these data from public databases, but now basically we use these block matrices. We put the stuff into block matrices, and then we do the same sort of thing, this sort of optimization, again, multiplicative update rules, consensus matrix, and so forth. And I'm sorry, I'm almost running out of time. But some results that, for example, we find 14 disease associations that are currently not present in disease ontology, for which we find evidence of their relationship through comorbidity data and literature curation. Again, genetic interactions for human, even though they're very small, they were the most important predictor, because again, we tested how well the fusion works with and without certain data, but omission of, so basically every type of data contributes to the quality of the model. And interestingly, 80% of disease ontology we can recover from fusing all of these types of data, so there is still a room for improvement. And lastly, I'll just gloss over this in a couple of slides. To do multiple network alignment, we know that it's much harder than doing pairwise network alignment, and it has to be heuristic. So in our recent bioinformatics paper from a few months ago, we used a similar methodology to align the 5 largest PPI networks that we have so far: human, Drosophila, mouse, C. elegans, and yeast. So again, we use block representation of all of this. This is just an illustration of from human to everybody else: R12, R13, R14, I14. But everything else is there. This is R2, 5, and so forth, right? And this is, again, to illustrate that basically you're sharing this G1 over all of these, and this is how you fuse all of these data. Now, here an interesting thing is that these R1, 2, and so forth, R2, 3, and so forth, they come from sequence similarities. So you put in sequence similarities, but then you integrate them by imposing penalties from the topology of these data. But now, even once you do your model and you reconstruct these, basically, so now based on the alignment, basically based on the sequence similarity amongst all of these 5 networks and topological fusion, topological similarity of all these PPI networks, you come up with these scores. These are reconstructed RIJs. But then you still have to address maximum weight K-partite matching problem. This is 5-partite here, which is NP-hard. K-partite matching problem is NP-hard already for K equals to 3 or more. So we have a small heuristic here, basically how you solve that, and it deals with merging data sets in some specific order that gives you the best performance. And in terms of results, only quick remarks that from sequence alignment, so this is basically top scoring pairs of annotated proteins in millions, and this is percentage of functionally consistent pairs. From sequence, you see the dashed lines are from sequence, and the solid lines are from basically this data fusion approach. For biological processes red and molecular function is blue. And when you compare to other methods, basically you want to find these clusters that are basically in the same cluster. You want to have genes from all species, right, that are aligned based on this collective evidence. So the more species you have, the genes from more species in the cluster, the better. So this is 2 species in a cluster, 3, 4, 5. So blue is good. And this is fused. This is our multiple network alignment. And you see that compared to other methods, we have fewer clusters, but they are basically they contain nodes from all 5 species. Then over here is the number of proteins in those clusters. Even though we have fewer clusters, we have just as many proteins as, for example, beams and other methods. So coverage of fuse is really nice. And also in fuse, we have the most biological process consistent clusters and molecular function consistent clusters. This is fuse, this is fuse, this is competitors. And basically this is from all, so you have clusters of these genes from all species that are biologically consistent based on the evidence from sequence and topologies of all these networks. Okay, so just to summarize, we've seen one methodology, others are coming. How to deal with data fusion, like many of the speakers have already addressed. We've seen just 4 examples of the same methodology that is fairly versatile and I think can be very useful, and there is still more to come. We still have so much more data to come, and we will need new tools, and this is very nice, exciting area to be in. Just to thank the funding agencies, my group members, some current, some past, and to announce that I've just moved 2 weeks ago to University College London, and I started my full professorship there, so I'm starting a new group and looking for new postdocs and PhD students. So if you have somebody to send me, please do so. And thank you very much for your attention. Yes.
So in all these formulations, you have sum of terms, each of which correspond to a different data type.
Yes.
I find it very surprising that there are no coefficients. So you give the same weight to each data type.
Yes, that's very nice. You can do that. Yes, you can. Here there is a little coefficient. I didn't focus on that gamma. And this gamma basically is how much you get from sequence and how much weight you want to give to topology. And it turns out that 0.7 gave the best result. 0.7 contribution of the topology. So this is the topology, obviously, here. So you can do this. Previous ones, no.
Play with this problem?
This is kind of a future research thing. One of the students is working on that. Yes, but we didn't so far. Yeah.
So the J1 for the tree factorization with two data types, patients, genes, and drugs. J1 was the driver gene somehow. Right? So here, what is the J in this one? So you basically turn everything to one network.
You mean this block matrix? Yeah.
The J, what does it represent?
J is you're minimizing this sort of thing. Yeah. And everything is inside of these block matrices.
There's a common factor between all the networks.
In each one of these, it boils down that you are approximating this R12 with the proper thing there.
Do you have an interpretation for this J?
J is just not, you mean G or J?
Sorry, G.
G, yes. G are always, so basically, you mean in this particular example?
Yeah, in this particular.
Interpretation of G, okay. Right, this one, right here, okay. Right, so this is, well, let me work, I don't know. Yeah, it's basically from human. This is from human to all of these other guys, right? So these are sequence similarities, and then we want to reconstruct these based on this and the topologies. So G1 times G2, G3, and so forth. So yeah, this is the clusters. Yeah, the clusters. Hmm? Yes.
What is the interpretation of that?
You want these sorts of things. I think it might be easier if you look at it on other examples. So let me.
It's a metric that is shared by all the networks.
Yeah, but there is a real interpretation of them always. Like, for example, in this particular one, this is patient groups. It depends what you are clustering. Sometimes it's patients. Other times it's going to be driver genes or like genes, clusters of genes, right? And you'll be looking for the ones that, so it just depends on what data. Here it's easier because they're different types, so it's easier. Over there, they're all genes, but from different species.
So I think we'll take the rest of these over break. And let's thank them again.
All right, thank you.